Hello everyone. Today we're going to talk about a very important topic, that is Krebs cycle. Now, Krebs cycle takes place in the mitochondrial matrix, just like link reaction. We need to understand the fact that this cycle can only continue if this acetyl-CoA molecule enters from link reaction. If it is not for the presence of acetyl-CoA, Krebs cycle cannot proceed. So the very first step in this cycle is where a 4-carbon compound, which is oxaloacetate, is being converted into a 6-carbon compound, which is citrate. So these 2 carbons which are being added are coming as acetyl group carried by coenzyme A. Once coenzyme A delivers the acetyl group to oxaloacetate to be converted into citrate, coenzyme A goes back into the link reaction where it brings another acetyl-CoA molecule. Further, this 6-carbon citrate is being converted into 5-carbon and 4-carbon intermediates. The names are not mentioned here for all these intermediates, as it becomes very difficult at this level to remember all the names. Now, in both of these processes, where a 6-carbon is being converted into a 5-carbon and a 5-carbon is being converted into a 4-carbon, you can see that carbon dioxide molecules are removed, which is what we call decarboxylation. And we already know that wherever decarboxylation is going to occur, dehydrogenation is going to occur as well. So you can see, due to dehydrogenation, reduced NADs are formed in these 2 steps. After that, this 4-carbon intermediate is converted into another 4-carbon intermediate. Meanwhile, a molecule of ATP is released. This release of ATP or production of ATP is considered as substrate-linked phosphorylation. What is substrate-linked phosphorylation? That you can check in my other videos, where we differentiate between substrate-linked phosphorylation and oxidative phosphorylation. Okay, after that, another molecule of reduced FAD and reduced NAD are produced, and of course, these can only be produced because of dehydrogenation. So dehydrogenation is occurring in both of these steps, and that is how reduced NAD and reduced FAD are produced before the last 4-carbon intermediate is going to regenerate the oxaloacetate. This is how the entire cycle keeps on going. Now let's look at the products. So the products per pyruvate molecule is 3 reduced NADs, 1 reduced FAD, 1 ATP, and 2 molecules of carbon dioxide. Now here carbon dioxide acts